Hello, everyone. Welcome back to my YouTube channel, and today I am going to teach you about the ubiquitin-proteasome pathway. In eukaryotic cells, the major pathway for the selective degradation of cytosolic and nuclear proteins employs a molecule called ubiquitin. Ubiquitin is a 76 amino acid polypeptide that is highly conserved in all eukaryotes. When attached in chains to proteins, ubiquitin targets the proteins for rapid proteolysis. A ubiquitin molecule is attached by its carboxy-terminal amino acid, which is a glycine, to the amino group of a lysine in the protein of interest. In turn, this first ubiquitin molecule is attached by one of its lysines to the carboxy terminus of the next ubiquitin molecule. The ubiquitin-proteasome pathway requires a number of players in addition to ubiquitin and the target proteins. Several types of enzymes, designated E1, E2, and E3, work together to add ubiquitin to proteins. Finally, a structure called a proteasome recognizes a ubiquitinated protein and degrades it. Ubiquitination is a multi-step process. First, E1, which is also called a ubiquitin activating enzyme, activates the carboxy terminus of ubiquitin by a reaction with ATP. The resulting ubiquitin AMP then splits in a reaction that attaches ubiquitin to E1. In this way, the energy from ATP hydrolysis has allowed the attachment of ubiquitin to E1. E2, called a ubiquitin conjugating enzyme, and E3, called ubiquitin ligase, work in concert with E1 to add ubiquitin to target proteins. Most cells contain a single type of E1 but have several types of E2 enzymes and a large number of distinct types of E3 enzymes. Different E3s recognize different substrate proteins, a process which confers specificity to the ubiquitin-proteasome pathway. A particular E3 recognizes and binds to a particular target protein as well as an E2 enzyme. Ubiquitin is transferred from E1 to E2 and from there on to the target protein held by E3. This process occurs multiple times to produce a chain of ubiquitin on the target protein. Such polyubiquitinated proteins are recognized by their ubiquitin chains and degraded by a proteasome, which is a large, multi-subunit protease complex. The process of engagement with the proteasome and protein unfolding require energy from ATP hydrolysis. Inside the proteasome, the target protein is degraded into small peptides. Ubiquitin is released in the process, so it can be reused in another cycle. The stability of many proteins is determined by whether they become ubiquitinated. I hope this will be helpful to you. Thank you for watching, and don't forget to subscribe to my channel for more videos.